Good afternoon or good morning, depending on where you are, to everyone, and welcome. My name is John Draper. I am the Vice President of Research and Training here at the Stem Cell Network. It's a real pleasure to open today's Rare Disease Symposium, Preparing for Clinical Trials. This is the second Canadian Rare Disease Symposium that we have hosted, which we are again proud to co-host in partnership with our good friends at the National Research Council of Canada. As we gather virtually, I would like to begin by acknowledging the land that we each stand on as we meet today on a virtual platform. The work of Canada's Stem Cell Network and the NRC occurs from coast to coast and occurs on the traditional, ancestral, and unceded territories of many Indigenous peoples and nations, including the Anishinaabe Algonquin Nation, where Stem Cell Network is headquartered. Stem Cell Network and NRC honors, recognizes, and respects these nations as the traditional stewards of the lands and waters where we work and meet. So for those not familiar with Canada's Stem Cell Network, we are a not-for-profit organization funded by the federal government of Canada that supports stem cell that supports stem cell and regenerative medicine research, training for the next generation of highly qualified personnel, enabling knowledge mobilization of research, and enhancing the commercialization readiness of stem cell and regenerative medicine innovations. So on to today's program. The symposium is focused on two critical elements advancing therapies for rare diseases. Session 1 will focus on how gene therapies for rare diseases fit within Canadian and international regulatory processes. And session 2 will focus on profiling cGMP manufacturing capabilities here in Canada that are essential for bringing these therapies to clinical testing. So this reflects on truly promising progress in the gene therapy field in recent years, such as the single patient gene therapy trial for SPG50 at SickKids Hospital in Toronto, which halted disease progression and showed early signs of improvement. A powerful reminder of how research can translate into hope for patients. So by bringing together regulatory leaders, Canadian developers, and international partners, this symposium highlights the global considerations required to advance rare disease therapies and underscores Canada's growing biomanufacturing capacity as a key driver in moving these therapies from the lab to the clinic. Before we begin today, I'd like to just thank my NRC colleagues, Freya Verkoturen and Rossini Waratin, and my SCN colleague, Fiona Cunningham, for their leadership and contributions in shaping the symposium and building today's agenda. And just a couple of housekeeping notes. The symposium, which is being recorded and will be posted on SCN's YouTube channel, will run in 2 sessions with a short break in between. Your questions are encouraged for both sessions, and you can enter them through the Zoom Q&A box at the bottom of your screen at any point, and these questions will then be addressed at the end of the session. If you see a question that has already been asked, you can upvote that instead of submitting a duplicate. Finally, a link to the event booklet has been shared in the chat, and there you'll find today's agenda, the speaker bios, and details on Canada's cGMP biomanufacturing facilities. And now I'll turn it over to my NRC colleague, Freya. Thank you, Freya.
Thank you, John, for the introduction. And thank you and Fiona at Stem Cell Network so much for your tremendous effort for co-organizing today's event. Welcome, everybody, to today's symposium, and bienvenue à toutes et à tous. My name is Freya Verkoturen. I'm the manager of the Cell and Gene Therapy Challenge Program at NRC. Currently, there are 11 challenge programs at NRC, and each of these 7-year programs focuses on important aspects of our society, including artificial intelligence, aging in place, ocean research, etc. And in the context of our cell and gene therapy research program, the NRC researchers and our collaborators have re-engineered a gene therapy for a rare disease that is prevalent in Canada. And we have developed CAR-T cell therapies for blood cancers and for solid tumors. And one of these CAR-T therapies is currently being evaluated in phase 1 clinical trial. In addition, we have developed a large number of tools and platforms to significantly reduce timelines and costs for the production of cell and gene therapies. And these technologies are now available for licensure or distribution to Canadian and international partners, so that eventually these effective cell and gene therapies will be available and accessible to patients. Alors, la première session de notre symposium d'aujourd'hui se focalise sur les processus en place dans de diverses juridictions pour l'approbation de thérapies géniques, plus spécifiquement pour les maladies rares. En effet, des experts provenant du Canada, de l'Europe et du Japon partageront aujourd'hui leurs perspectives sur les processus et les délais, des défis, mais aussi des opportunités pour l'harmonisation internationale pour ces processus réglementaires. Cette session sera modérée par le monsieur Patrick Bedford. Patrick est le fondateur de We Can Reg Consulting Group ainsi que Vice President chez Morphocell Technologies. Il possède d'amples expériences en approbation réglementaire pour les thérapies cellulaires et géniques, plus particulièrement au Canada. So today our panelists are Dr. Tunekti from Health Canada, and Dr. Maruyama from Pharmaceuticals and Medical Devices Agency in Japan, Dr. Reichel, Chair of the EMA Committee for Advanced Therapies, and Dr. Satyamurthy of Aspire Bioconsulting. And this group brings global insights on regulatory frameworks and practical guidance for advancing therapies to the clinic. À cause du décalage horaire entre le Canada, l'Europe et le Japon, le docteur Maruyama nous a fourni une présentation préenregistrée que nous allons inclure dans notre première session. The second session today will be moderated by Dr. Kelly Perato, who is the R&D Director in Bioprocess Engineering at NRC. And during that session, various Canadian entities will be presenting their GMP-compliant manufacturing capabilities for gene therapies during 5-minute flash talks. As John mentioned, further information on their manufacturing capabilities will be available on the Stem Cell Network website. So thank you for joining today, and please join me in welcoming Patrick Bedford to lead today's first session.
Hello, everyone, and thank you, Freya, for that introduction. And thank you to Stem Cell Network and Canada's National Research Council for inviting me to moderate the first half of this session entitled Preparing for Clinical Trials for Cell and Gene Therapies Targeting Rare Diseases. We will be focusing on providing international regulatory insights for this first session. And I want to make sure that my goal will be to make sure we stay on time and to get to your questions at the end of this session. So what some of you may not know about me, in addition to the introduction that Freya provided, is that over the past 5 to 10 years, I think I've often been the first person in Canada that many global companies speak to when they begin to explore whether they should conduct studies in Canada. And I am unabashedly a Canadian advocate who has worked really hard in the emerging biotech regulatory space for about 20 years, half of which I spent at Health Canada, writing policies for transplant materials and cell and gene therapies. And I really do believe that part of a global strategy is to explore jurisdictions like Canada and elsewhere where you can accelerate some of your plans to get products to patients in trials and then market access. So for this session, just a quick overview. Our agenda, as you can see, has, and as Freya and John mentioned, has 3 speakers from different regulatory jurisdictions who will be providing 15-minute presentations about the Japanese PMDA, or Pharmaceutical and Medical Devices Agency, from Health Canada, and then from the European Medicines Agency. And then we will be hearing from a regulatory consultant who has niche experience in rare diseases. And Sam will be providing practical insights into how you might digest and apply some of the information that you'll be hearing today from the regulators before we have our panel discussion and question and answers. So after those presentations, we should have about a half hour, of which about half of it I will be leading with some questions that I've prepared ahead of time. And then we'll be getting to some of the questions that you should be typing into the question and answer panel, and Stem Cell Network will be helping me to prioritize. Ultimately, our goal today is to provide practical jurisdictional insights. And I think we will have succeeded at the end of this session if you even consider a more nuanced global regulatory strategy for some of the things that you might be developing, other than just going to a single jurisdiction based on target available market size. So that's my goal. And when I say I'm a Canadian advocate, that's what I truly hope. So before introducing our first speaker, I just did want to provide some basic observations for context. Most early stage global companies that I speak to have plans, and they base it on real or perceived belief that investors only care about the US jurisdiction. All of the market plans, the development plans, everything use US FDA terminology. And you'll note that we don't have an FDA speaker at this particular session. We will be hearing from other key jurisdictions. And I think you should learn more about those jurisdictions. So we've assumed that there are many of you who are already familiar with the US FDA terminology. I'm talking, and the process, the pre-submission process, the submission process and terminology. You hear terms like interact meetings, pre-INDs, IND enabling studies, submitting INDs, but there's other terminology and other processes out there. Some of you may already be aware of and carefully have read even this month, a new draft guidance came out from the US FDA on guidance for innovative trial designs for cell and gene therapy in small populations. But most of you may not be as familiar with the European Medicines Association or some of their member state requirements, Health Canada, or the Japanese PMDA. So I'm really happy that we got some great speakers from those jurisdictions. I do want to highlight all of those jurisdictions are full members or founding members of the International Council for Harmonization, or ICH. So I'm going to get out ahead of them and say any research you do in one of those jurisdictions is closely harmonized with the US FDA and meaningful. And if you gather data there, it's pretty portable. And that's an important thing to note. So they're all highly relevant regulators. They work and harmonize global regulatory expectations. And I think we'll see a lot of similarities, and more similarities than differences from our presenters today. And I strongly suggest that it's worth it to you to explore whether you can do some of your development work in those jurisdictions to get to the clinic and the market faster and more efficiently. I think you'll agree that we should work to meet the needs of small disease populations across the globe earlier. And finally, I think that speaking with these jurisdictions in early development will de-risk your clinical translation journey and gather more valuable insights along your way, and I encourage you to do so. So without further ado, I will be turning this over to Dr. Omar Tunekti, who works at Health Canada, and I've known him for years. He's a familiar face to me and to many of our Canadian audience because you will see him at early stage pre-submission meetings with Health Canada, focused on cell and gene therapy, where he provides advice as a manager of gene therapies and radiopharmaceuticals in their review division. He's got a lot of really good review experience and policy understanding. So I really look forward to your presentation, Omar. And if you don't mind throwing on your camera and walking us through your slides, we will try and keep you on time, about 15 minutes, and then we'll get back to some questions on our panel. Thank you very much.
Thank you very much, Patrick, for the introduction. And first of all, thank you so much to the organizers for the invitation. It's a real pleasure to be here with you all today. So this is the outline of my presentation, and with three key themes. What's expected in the clinical trial application here in Canada, the areas of flexibility for rare diseases or gene therapies for rare diseases, and the expected expedited review options. We are part of the Biologics and Genetic Therapies Directorate within Health Canada of the Health Products and Food branch. And my team is responsible for the review of chemistry and manufacturing information for cell and gene therapies. And the clinical review is covered by, at the clinical trial stage, by the Office of Clinical Trial within the Center for Vaccines, Clinical Trial and Biostatistics. And at the marketing stage, it's going to be, depending on the indication, can be in the different centers, can be in our center if it's oncology or hematology. So that's our organizational structure. In Canada, unlike the US or in Europe, we don't have a dedicated orphan drug designation or legislation, sorry. Instead, the rare disease drugs are reviewed under the same regulatory framework as other drugs or biologics. but we do have, we apply flexibility in evidence requirements where appropriate. And all gene therapies are regulated under the Food and Drugs Act and regulations. And Health Canada's oversight covers most of the product life cycle, starting with the clinical trials, as you can see on this slide, the market authorization, post-market changes, and also some post-market surveillance. Clinical trials for rare diseases in Canada follow like similar clinical trial process as for biologics. So the requirements are listed in the Division 5 of our regulation. But we do, as I mentioned, apply some flexibility because of for example, the small patient population. So the sponsors are required to submit a clinical trial application in Canada before starting the trial. And we review the information within 30 days. Therefore, there is no cost. The information that's requested in the application are high-level description, the protocol, clinical trial protocol, the investigation brochure, informed consent, and chemistry and manufacturing data. So in the next slides, I'm going to cover more the information expected on the chemistry and manufacturing side, and focus maybe on more the gaps that we noted during review of some of the clinical trial applications for gene therapies. So I hope that most of you know that there is a standard format, which we call the CTD, Common Technical Document format, to submit the information. And the Health Canada requires that the product meets the GMP and demonstrate quality, safety, and consistency. The gene therapies are in general made with using a complex manufacturing process with multiple critical starting material and ancillary components. And we recommend that each of these materials has a dedicated drug substance module in the file, rather than grouping all these components under section 3.2 S23. This covers also some of the, for example, for cell therapies, the apheresis material, and it's important to include information about the chain of custody or identity, and specifically also, this is something that's sometimes missing, the hold times for all these products and the stability data to support those. Another important element that's key for the cell gene therapies is the use of human serum albumin as an excipient. And this is a reminder that this is, as part of our regulation, require that the serum albumin is sourced from the Canadian market. So review of these, the clinical trial is mainly focused on safety, and in general, the sponsors, we know that the sponsors invest most of their effort on the efficacy piece to demonstrate that the product works, and we all hope that it works. But as a result, we often receive clinical trial application with important information related to safety is missing. For example, in terms of the raw materials, we do not receive information about all the components, for example, in cell culture media or in solution. It is important to identify those who are of biological origin, and in terms of safety, it is important to provide the certificate of analysis of origin to demonstrate that the product is safe from BSC or TSC risk. In general, also the control strategy for us is important because it's tied to the safety. So any changes related to that, to the control strategy, for example, the changes in the acceptance criteria of some of the release and stability testing should be reported as an amendment for approval by Health Canada. I think it's important that any details or any information related to the structure of the file or important information or strategy that you would like to share with the reviewers be included in section 107. I think it's an important section where you can communicate with the reviewer and explaining some of the details of the submission. It would be super helpful for reviewers to proceed with their work and also avoid any back and forth in terms of question answers. In terms of areas of flexibility, we understand that there is reduced batch data at the submission. Limited data batch scale, commercial batch scale, or batch analysis data is accepted. Same for stability data. Some limited stability data can be accepted. Again, back to section 1.07, or in the appropriate section, you can justify why there is limited data and why these data support the proposed shelf life. Process validation, same, there is flexibility there, and also in terms for the specification. So we allow a bit of wide specification, but we expect them to be tightened as the knowledge and the product progresses through the life cycle. One of the very important areas for gene therapies is the comparability. And the limitation of the size and availability of the lot. So we allow also the use of platform knowledge or prior experience to justify the comparability assessment. We, in general, align, as Patrick mentioned, with the ICH guidance documents. I'm going to cite specifically the quality by design, and that's the recommended approach to document and acquire knowledge about the product. But we also can leverage any assessment done by our partners in Europe or other jurisdiction. And we will use also collaborative review with US FDA or EMA or with our access partners. Just to, this is not my expertise, but on a clinical side, as you can see on the slide, there is also some sort of flexibility because of the small population. So we accept smaller single-arm trials, use of surrogate points, and adaptive clinical trial design. My last slide is around the accelerators for development, and you have two main ways to do it. The first one is the priority review. This is more at the marketing stage. So what's key is, as you can see the details here, it's going to shorten the review time from the 300 days for a new drug submission. That's the standard review time for us. So from 300 days down to 180 days. So if the drug provides significant benefit, the notice of compliance with conditions, that the second route that you could use, and it shortens the review time from 300 to 200 days. but it's going to be based on the promising evidence, for example, for a phase 2 clinical trial. I think what's my last point is around the communication. So please talk to us. I put in there the pre-submission meetings, either preclinical trial or new drug submission or pre-market meetings. So we're open to help and answer any question with a specific product line or the regulatory submissions. But I think it's important to know that it's essential that sponsors to make best use of your time and our time, that you come prepared with well-defined product specific questions. This is going to ensure the discussion is focused and adds value to both the sponsor and us as reviewers. We typically provide you with advanced feedback, so like written responses to your questions. These meetings are not intended for general regulatory education or to high level type of filing. So if the sponsor requires fundamental guidance on submission planning or dossier or application preparation, they should engage a qualified regulatory consultant before requesting a meeting with us. With that, I'm going to be very happy to answer any question. Over to you. Thanks.
Thank you very much, Dr. Ginecti. I think, first of all, I think that was a wonderful overview and provided some very good references to Canadian regulatory and international regulatory guidance. Obviously, this is being recorded and will be available on YouTube. So if somebody missed some of the details, go back, look at that, look at some of the references. And what you did is summarize a regulatory context in 15 minutes. So that's pretty impressive. It is hard to figure out exactly what to go through, but I think very good job giving us a good summary. And just some key messages that came out to me to highlight for people: cell and gene therapies for rare diseases in Canada, regulated by Health Canada and in their Biologics and Radiopharmaceutical Drugs Directorate, where OMAR works. There's a clear pathway and it requires a special formatted submission of manufacturing preclinical data, and you have the references for even specific modules in a regulatory submission. And Health Canada is open, accessible, able to speak with people, and taking a flexible approach to rare diseases, and especially with products that are like cell and gene therapies. I will take a little bit of liberty and highlight and foreshadow a little bit, I think, that what's good for Health Canada is good for FDA, which is good for EMA. It bears approaching different regulators and having discussions with them to de-risk in those jurisdictions. But it is possible. Omar mentioned a pre-submission pathway. Preparing a briefing book for the FDA is very similar to using it for Health Canada. There's a lot of cut and paste involved. Yes, just Canada has a regulator, very good, and you have some of the contact information there. Now we're moving on to our next presenter. We have a presentation from Dr. Yoshiaki Maruyama. He's the review director of the Office of Cellular and Tissue-Based Therapies Products at the Japanese Pharmaceutical and Medical Device Agency, or PMDA. He's recorded his presentation because of time difference and won't be able to participate in our panel or in the Q&A, but we greatly appreciate his additional effort to contribute. And I encourage you to consider when and how you should explore clinical trials in Japan. So let's go ahead with that recorded presentation.
Good afternoon, everyone. I'm Yoshiaki Maruyama, Director, Office of Cellular and Tissue-Based Product, PMDA Japan. First of all, I'd like to thank the organizer for the opportunity to give this presentation today. Since we are in different time zones, I recorded my talk ahead of time. This is today's outline. I'll be introducing key considerations when initiating a clinical trial in Japan. When initiating a clinical trial in Japan, the applicant is required to submit the clinical trial notification to the PMDA, along with the following 5 documents. 1. I believe submitting such document to regulators and undergoing regulatory review is a globally standard process. However, the review periods differ by country. In the case of a new product, especially an initial notification, the investigation period is 30 days in Japan. For second and later notification, including those for other indications, the investigation period is 40 days. This slide shows the number of clinical trial notifications for cell and gene therapy since 2014. 2014 was the year that Pharmaceutical Affairs Law revised and an early approval scheme, conditional and time-limited approval scheme for cell and gene therapy product was introduced. A total 151 initial clinical trial notifications were reviewed by the PMDA. Japan's development trend has shown a consistent number of approximately 15 initial notifications annually since 2016, and there has been a growing focus on gene therapy. The largest proportion of development is in the oncology area. To prevent the occurrence or spread of hazard to the public, PMDA investigates the study protocol and other documents submitted with the notification for assurance of the safety of the study subjects. To reduce the inquiry from PMDA during the CTN investigations, engaging in the consultation with PMDA is recommended to verify that the investigational product meets the standards set forth in the relevant quality and safety guidelines before submission of the clinical trial notifications. Otherwise, PMDA won't be able to complete CTN investigation within 30 days. Until the investigation is complete, clinical trial cannot begin. In order to facilitate development of cell and gene therapy product, PMDA started consultation on research and development strategy by 2011. This is not simply giving the advice to applicant, but thinking together against challenges. In such consultations, advice is provided on the tests required in the early stage of development and the requirement for starting clinical trials. The table shows the number of the consultation for cell and gene therapy past years. In Japan, a fee is charged for consultation with the PMDA, but if certain requirements are met, 90% of the cost will be covered by the government. If consultation is held over multiple days, the fee for one consultation will be charged for up to 3 consultations. The Cartagena Protocol on Biosafety to the Convention on Biological Diversity is an international agreement that aims to ensure the safety handling, transport, and using of living modified organisms. The Cartagena Act is a law concerning the use of gene-modified organisms, living modified organisms, and covers the use of GMO, LMO as medical products in Japan. The Cartagena Act has two types of regulations. Type 1 use is the deliberate release, the use of GMO without preventive measures against their release into environment. Applicant planning to conduct clinical trial in Japan should submit Type 1 application to PMDA MHLW, Ministry Health, Labor and Welfare. It's necessary to obtain approval from the MHLW regarding Type 1 use at medical institution before enrolling the first patient in the clinical trial. In order to facilitate development of cell and gene therapy product, several improvements has been implemented, making the administrative process of GMO regulation rapid and predictable. The PMDA have published a considerable number of instructions, such as specific descriptions on environment risk assessment for adeno-associated virus vector to provide information about the Cartagena Act to future applications. The median of regulatory review time reached to 3 to 4 months for approval of Type 1 use. This is an example of the process up to the clinical trial notification. Depending on the development status, discussion can be carried out either sequentially or in parallel. EMDA provides checkpoints addressing the frequently raised issue during face-to-face consultations. As consultation materials are submitted as part of the CTN document, please utilize the publish checkpoint when preparing these documents. Next, I will introduce key considerations to keep in mind when moving towards commercialization. In Japan, 21 cell and gene therapy products have been approved at the moment. Among the approved products, 80% have indications designated for orphan disease. 2 conditional and time-limited approval products, collagen and heartseed, have been discontinued. The standard review period is set at 12 months. However, for priority review product designated for orphan disease, the review is conducted within 9 months. All review reports for cell and gene therapy products are published in English. Approved cell and gene therapy products are eligible for reimbursement by the National Health Insurance Scheme. Patient fees are maintained at a reasonable level, even by conditional and time-limited approval product. A fee ceiling is put in place when patient fees are very high. I'd like to introduce orphan designation system in Japan. For orphan designation, these 3 criteria, number of patients, medical needs, possibility of development, must be met. Number of the patients are less than 50,000 in Japan. This slide shows orphan product development support program. This program aims to deliver new, safe, and effective orphan product for orphan disease into clinical practice at earliest opportunity. To achieve this goal, NIBN offer financial support for the development of orphan designated product and also provide guidance and advice for obtaining marketing authorization. Next, I would like to introduce conditional and time-limited approval scheme. This is a two-step approval scheme. Products are granted first marketing authorization when efficacy can be predicted through exploratory clinical trial. However, in order to submit a second application for approval within the granted time period and demonstrate efficacy to maintain the authorizations, sponsor is required to conduct strict post-marketing surveillance as a condition of the first approval. Conditional and time-limited approval scheme is intended to enable early patient access to cell and gene therapy product whose efficacy has been predicted in the early stage of clinical trials. However, excessive expectation regarding efficacy may lead to misjudgment of hypothesis in post-marketing evaluation. In response to discontinuation of two products, operational improvements such as enhancing the predictability of the second stage approval are necessary. MHLW and PMDA is currently developing notification on handling of conditional and time-limited approval scheme. PMDA is strengthening the dissemination of regulatory information globally to promote of development in Japan. Please access to our website. This is the end of the slide. I look forward to future interactions. Thank you.
Excellent. So I have to say thank you to Dr. Mariana in absentia for providing an informative overview of the regulations and processes relevant to cell and gene therapies for rare diseases in Japan. I also will note that I see questions popping up in the background, so that's excellent. Please keep it up. We will get to those during panel discussion, and then we have some time at the end even for any other question and answers that we may have missed. And I think we're doing okay for time still, so let's keep that up as well. So next we have Dr. Ilona Reichel, who's a quality assessor at the Austrian Medicines and Medical Device Agency. And she's also the chair of the European Medicines Association, or EMA's, or EMA's Committee for Advanced Therapies, or the CAT. I must say I only just met Ilona leading up to this session, but I loved our candid discussions so far. And I don't think she'll be afraid to give us some candid insights, which I think she will carefully disclaim as her personal perspective, but we should listen carefully because she does have extensive knowledge in this space and experience. And so I'm sure we'll provide some valuable information for people to consider when they look into potential European Union jurisdictions. Without further ado, please go ahead, Ilona.
I hope my slides are coming up. Please let me know if this is the case.
Yep,
They
you're all good.
look good. Yep.
Okay. So good afternoon in your case, good evening in my case. I will tell you about the regulatory system in Europe, and doing that in 10 minutes is actually quite challenging. So I'm going to talk about clinical trials in the European Union or European Economic Area, and what that means after going through my disclaimers that I'm neither talking for the EMA nor for my national agency. The biggest thing to not an assessment.
Sorry, I think it's lagging and we're dealing with some technical issues. I don't know if it's just on my end.
Okay, if I have technical issues, I can try to switch to a hotspot. Hopefully that will be more stable.
Thank you, and sorry about that, everybody. I think despite all the best preparations, we still all deal with the IT gremlins.
Yes.
Your camera seems to be working well now, but it's no longer
yeah, occasionally it does. Occasionally the network is unreliable, and I will see how it goes. So you can still see my slides, or do I need to?
No, we have your camera, but no slides right now.
Okay. Right. So I hope it's back now.
Oh, yes, that looks great. Thank you.
So I was at the European Medicines Agency, which has a strategic and not an assessment role in procedures. It hosts committees and working groups, and we, the assessors from national agencies, come together in these working groups and committees to share assessments and make decisions. Also, clinical trials in particular are assessed by the national agencies, and EMA is not involved other than that EMA hosts the database that the trials are managed with. And the European Clinical Trial Framework is primarily based on a single EU regulation, and that is Regulation 536/2014, or CTR in short. Yes, this gives you an idea how, when you deal with 27 member states, things are not that easy. When it comes to regulatory convergence, you should probably talk to the Europeans because we have decades of experience with it. What you can see on the slide is that we have the legislator, which is the European Commission, being responsible for European laws. We have the national competent authorities, and the heads of the national medicines authorities form HMA, heads of medicines agencies. And
Ilona, it seems I think you've frozen again. We'll just give you a minute. I assume it's on, it's not just me. I'm sure Ilona will try and log back in. Oh, there we go.
I'm sorry. So
no problem.
I'm getting faster in returning. So the legal hierarchy in Europe is based on regulations where the legislative text immediately applies to. Yeah, I can switch my camera off. Maybe that makes it more stable. Where the text immediately applies to all EU member states. The older legislations were mostly directives, and directives needed to be first incorporated into national law before being applicable. Then we still have national legislation, hopefully that only affects very minor aspects. By this time, we have the EU standards, which are basically worldwide standards, the GXPs, so GMP, GCP, GLP, etc. And we have the pharmacopoeias, which provide, where the monographs provide binding text, certainly at the level of marketing authorization, whereas the general texts are more on the level of recommendations, but it's very pertinent to consider them even as you're going through development. And then we have legal and scientific guidance and Q&As at the bottom level. And for clinical trials, pertinent documents are summarized in EudraLex volume 10. So if you Google EudraLex, you will find 10 volumes. One is legislation, I believe. So there are different topics in different volumes, and volume 10 is for clinical trials. Information for the level of marketing authorization can be found on the homepage of the European Medicines Agency, which is here, ema.europa.eu. So what are the potential differences that you might not be fully aware of? I already mentioned that the European process is a harmonized joint process where member states retain the authorization step. That definitely adds time compared to the situation where, for example, in the USA, you have a single country, a single authority, nobody else who you need to talk to come to a joint decision. The guiding principle in Europe is the clinical trial identification number, and the data maturation is the developer's responsibility. And that is largely because you could be conducting your phase 1 trial in, I don't know, in Germany, your phase 2 in France, your phase 3 multinationally. So tracking the progress towards marketing authorization is the responsibility of the developer. Whereas in the US, the organizing principle is the product, e.g. the IND number, and with personalized products, for example, that I believe is creating current challenges because it's based on single entities. We do not have that issue in Europe at the moment. So there is no legal obstacle to personalized medicines. The other thing to consider is that although our process is longer, it does include ethics assessment. So for each member state, we have a single opinion, and that is including the opinion of the NCA and of the ethics committee. To my knowledge, IRB review is not included in the US. We do have stricter GMP compliance in that you need a manufacturing authorization prior to getting into first-in-human trials, and that also means you would be inspected if you are a European manufacturer. And in order to conduct a clinical trial in Europe, you would have to have a European importer certifying compliance with the requirements of GMP. Whereas in FDA, the GMP inspection is for sure at the level of the BLA, but usually early on, it's a paper review of the facilities as part of the IMPD, or of the IND submission, I should say. We also currently do not have a single process for clinical trials that cover more than one legislation. So in other words, a co-development of a medicine with a device or with an IVD. This is currently being addressed in the Combine project, and we hope to progress rapidly to enable these core developments better. Whereas you have the one-stop shop at FDA. A brief outlook on CITES, the database, which is the workhorse of clinical trials. It is the database in which we work together. There is an EU portal with secure access to sponsors, industry and academia. It's the same for everyone. For member states and ethics committees, obviously for the European Commission and for the European Medicines Agency who host the information. So all the information on all trials in Europe is in this database. And part of the database is accessible to the public. The website for CITES is given to you below. And it is a relatively recent introduction. It is mandatory since the beginning of 2025. If you realize the discrimination between the regulation being from 2014 and the database from 2025, that's approximately how long it took to develop this database that enables this complex interaction of all the various actors and member states. I've tried to draw a simplified model of how it works without giving timelines, because I think that goes beyond the scope of today, and that certainly would take more minutes than 10. So the sponsor submits via the CITES database, and the sponsor selects which countries or maybe only one country they want to involve in the clinical trial. As a first step, the RMS or reference member state is selected, and that is on the basis of the proposal by the sponsor. So in this case, the sponsor probably suggested Italy, Italy accepted, and Italy is now the reference member state, which means they are the one, Italy is the country that handles the majority of the workload. Then comes a validation step, formal documentation, whether everything is available, and each country can, again, the lead is Italy, but each country can also add deficiencies if they spot one. And there is a round of information requests if needed. When validation is complete, which also includes the decision or the formal acceptance that it is a clinical trial according to our legal requirements, we go into the assessment step. And the assessment step is divided into a shared assessment and a national assessment. The shared assessment, and I will go into that in the next slide, is also called Part 1 and involves documents such as the protocol. And in this case, Italy would conduct the assessment, write an assessment report, and other countries can comment on this assessment report and add additional comments if they have some. Therefore, we need a consolidation step at the end where the list of deficiencies, and it's important to note that these are deficiencies that are potentially blocking. So that's not, shouldn't be nice to knows. Those are really blocking deficiencies. And there's the possibility for an information request. Again, the time for that is very short. And then there is decision, which will be entered. And I now assume that in this particular case, the decision is positive. The other one is the national aspect, which every country looks at for themselves. And this also has the possibility for information requests, leads to a decision, and if both of these, Part 1 and 2, are positive, the trial can go ahead. It is also possible to submit Part 1 separately prior to submitting Part 2 at a later stage, but I'm not going into these details now. The information is then partially made public. And so in summary, it's a single procedure which does incorporate ethics committees, which has shared and national aspects. And Part 1 conclusion and Part 2 conclusion result in a single national decision. So each member state separately would provide their authorization letter for the trial. And fees are payable nationally and adapted to the role that the respective country plays in the overall assessment. Now, I talked about Part 1, Part 2. What are the shared documents? The decision of whether it's a full clinical trial or a low intervention clinical trial, we can talk about that later. The protocol, the investigator's brochure, the investigational medicinal product dossier or the auxiliary medicinal product dossier, or it used to be called non-investigational medicinal product dossier or NIMPS, documentation of EU compliance and labeling. And national aspects would be informed consent, compensation, recruitment, data protection, suitability of personnel, suitability of trial site, damage compensation, so insurance, and compliance of collection, storage, and future use of biological samples. And so there is the option to still submit a mononational trial, of course, but the procedure really was designed to enable a better and more harmonized assessment of multinational clinical trials. Transparency is a big thing in Europe, and the approach is public by default, which means that comprehensive public disclosure is required and facilitated via CITES. What's definitely published is the trial protocol and the IB, clinical summary report and study reports for the scientific community, informed consent forms and lay summaries for the public. There is a staggered approach for the publication of proprietary preclinical and clinical data, but it does occur. And the only things that are not disclosed are the proprietary manufacturing details, i.e. the quality IMPD. And again, you see the link or the website for the transparency rules that are applicable. And so I can summarize, the EU clinical trial process is governed by a harmonized European legislation and is a joint review by concerned member states. So in other words, if your trial occurs in Austria and Germany and France, those are the countries involved. Spain would not be involved if it's not part of the trial. It does incorporate ethics review. It's managed through CITES. We do have stricter GMP requirements compared to other regions. That, I believe, is also largely due to our history. Patients in severe disease are considered vulnerable and are specially protected rather than reduced requirements. There is no legal obstacle to personalized approaches, and relevant information on clinical trials is on the EudraLex Volume 10 webpage. I did not include any information on scientific advice, but we obviously have that. We have it on a national level, we have it on a European level, we have innovation networks. So maybe we can go through that in the discussion afterwards, but I didn't include it in order to stick with the timeline that I had for presenting the European system. So thank you, that's for me.
Wow, that was wonderful. Thank you very much, Dr. Reichel. It's very important and helpful to demystify the EMA and member state activities. So thank you. That was great information. And I know you worked through IT challenges and we're still just about on time. So thank you very much. We do have a couple minutes over, but we'll be able to deal with that in the panel discussion. So, and I have to say, I particularly liked, in addition to your last slide, which I enjoyed, slide 6, where you highlighted European and American similarities and differences, very helpful. I'd love to add Canada to that at some point. I'd squeeze it in the middle. And I think we may hear a bit about that from our final speaker now. So I will be introducing Dr. Sarmitha Savyamurthy. I know her as Sam. She's the president and principal consultant for Aspire Bio. I've known her for about 5 years, and Sam is a fellow regulatory consultant with niche focus on emerging biotechnologies, and one of the few qualified regulatory consultants in Canada who I'd actually refer some business to when I'm too busy with consulting. So that's high praise. I have a high standard for that. And she'll be speaking about similarities and differences in global regulatory expectations. I think it'll be a little bit of a different flavor than hearing from the regulators. This is, as you interpret what they say, what does it mean? And I think as a consultant, Sam will be able to tell us a little bit of what she learns from seeing across multiple different jurisdictions with a single client. So without further ado, Sam, please pull up your slides, and I look forward to hearing your presentation.
Thank you, Patrick. And I want to thank the previous presenters before me, because they did a phenomenal job outlining what the process looks like from each of their jurisdictions. And as Patrick said, this is going to be a little bit of a different flavor, a different color, a different lens, because it's thinking about what the global strategy for a rare disease may be as we embark on from proof of concept to first in human and then eventually commercialization. So the aim here is to design a global regulatory strategy whenever possible. And I think it was Omar who brought this up, that the regulators are an invaluable resource, but ultimately the company does need to come up and develop and be responsible for their own regulatory strategy. At the end of the day, each individual regulator or regulatory body is not going to know what the drivers are behind the company decisions. And so that's all going to flavor what that regulatory strategy is going to be. And the regulator can and will offer feedback on your strategy, but they cannot be expected to tailor a customized roadmap for an individual company. And so there's a very big difference between going in your submission or your pre-submission meetings saying we intend to do this and do you agree with this, versus here's what we hope to aim to achieve. How should we go about doing that? What should we do? And so the latter part of this, a regulator will not be able to provide that feedback. And to understand the jurisdictional similarities and differences, Ilona did a great job comparing EU and FDA. And as Patrick said, it would be great if somewhere we can build a comparison across all jurisdictions, but that would be a very big chart, I think. But to understand the jurisdictional similarities and differences in the areas that you're interested in when planning your global regulatory strategy. And so my intent here is to kind of go through the different areas of consideration. So the non-clinical study design, the CMC strategy, the clinical study design, and then ultimately the regulatory strategy and how that should fit in. So looking at it from a non-clinical consideration, safety cannot be avoided. If something is going to get put on clinical hold, it's going to be because your safety is in question or your quality of the product is in question, really. Often it's not because the idea was a bad one. So the safety cannot be avoided, and it's essentially a similar requirement across all jurisdictions. However, what that looks like for a rare indication from jurisdiction to jurisdiction is going to be different. So for example, given the indication and maybe the route of administration, are NHP studies needed? If so, is that a GLP or a non-GLP NHP study? And what does the GLP tox study necessarily need to look like in terms of model organism, perhaps the size of the study, et cetera? And so this is information that is going to be supported by the company to justify what they want to do in the non-clinical safety assessments. And sometimes this can be the biggest risk that you have to a program, because some of these non-clinical early work, like maybe your early efficacy information or the non-GLP tox assessments, may already be underway or done by the time a company is really thinking about their pre-submission meetings. And so you really want to think about and talk to your regulatory team and or a regulatory advisor if you don't have a regulatory team about when you should have that pre-submission meeting, because the pre-submission meeting timeline is going to be driven by what you're identifying as the biggest risk to your program. And sometimes that biggest risk to the program is the non-clinical study that you're going to design. And part of this assessment is to consider the type of material that you're going to be using in your non-clinical studies. So there has to be some sort of an assessment made to ensure that the material that you're using for your non-clinical work is comparable to the clinical material, and that comparability assessment can look different based on the different jurisdictions. So having these conversations early on and ensuring that the regulator that you're speaking to understands how you're thinking about your program is going to be critical. Now, Ilona kind of stole my thunder here with this, with her comparison slide, but essentially from a CMC perspective, this can look very different jurisdictionally. So for example, as mentioned, EU, you must have a certified GMP facility that is manufacturing your clinical material. For the FDA, you need to be GMP compliant, but no inspection is required. That facility doesn't need to be certified, but they have to ensure that they're following GMP principles. But FDA is really clear that it doesn't need to be full 21 CFR Part 210 or 211 compliant, which means it's a phase appropriate GMP. Health Canada, GMP compliance is needed, and the control of the process and release of material is scrutinized. And when we think about Australia, there's no GMP requirement, and often it's not TGA that's reviewing your package, it's by the individual IRBs. And so thinking early about what your overall global strategy is going to look like is going to deeply impact who and how you're manufacturing. Because especially in the rare and ultra rare disease space, you're not going to manufacture that many clinical lots for the entirety of your clinical trials. Perhaps it's only one lot that you're manufacturing. And so if you want to consider the different jurisdictions, it's crucial that you're thinking about where you're going to end up with that material and how that material should be made. And so again, engage your regulatory team and or advisors early in the process before you finalize your manufacturing strategy, because that information is going to feed into who and how you're going to be manufacturing that. Also, there are other nuances to think about, especially in particular for the rare disease space, because if that first lot is potentially going to serve as your pivotal trial material, then that lot has to be released in a particular way. So for example, what is the potency strategy? And this is going to have jurisdictional differences. So is the potency, take AAV for example, is an expression assay sufficient? Do we need a functional assay? Should it be a relative potency assay? And is that assay going to be fit for purpose, qualified, or validated? So all of this, especially the potency strategy, should be discussed with the jurisdiction that you're going to be be doing your trial in. Now, in terms of the study design consideration, first in human is very similar across the jurisdictions, I would say. You have to understand what the standard of care is in the country that you're in and how your proposed therapy fits into that standard of care. And is there a concurrent medication that may impact your study? And if so, how are you going to deal with that? Now, this is for first in human. I think one of the things to consider here is that getting to the clinic is not the endpoint. Getting to commercial is the endpoint. Making that drug available is the endpoint. And so thinking through the study design is also jurisdictionally impacted, because perhaps you can have a phase 1-2 study, perhaps you can take a look at some sort of an accelerated approval pathway. And so this should be also part of the consideration and discussions at that pre-submission meeting as to how you're thinking through your clinical design. From a regulatory perspective, understanding the regulatory pathways in the different jurisdictions is going to be paramount in this decision making. So what are the pre-submission meeting options that are available to you? And once you have that pre-submission meetings, can you have other meetings after that? And how many meetings can you have before you have to submit? Or not have to, but you run out of meetings. And is there a cost to the pre-submission meetings? Often not, but sometimes some of the jurisdictions do have a nominal fee associated with the pre-submission meetings that needs to be considered. And so as a company, sitting down and thinking about what are your drivers when choosing the jurisdiction, sometimes this, as Patrick mentioned, perhaps it's always looking at the US or another country, but what are the drivers for choosing that jurisdiction should be an active conversation that a company is having. Is it cost? Is it time? Is it ease of filing? Is it the knowledge that the executive team or the team might have with regards to that jurisdiction and how it functions? And definitely patient population should go into this as well. Canada is a smaller population, but we have these founder population pockets where rare disease may not be so rare in a particular area within Canada. So that's a compelling reason to do some of these clinical trials in Canada. And then what are the different program designations or special pathways that are available for rare indications? Perhaps that's part of the decision making as well. And so my final slide, my closing thoughts, if there is no other message that I convey than this one, it is to take advantage of the pre-submission meetings. There is no obligation to submit once you have that meeting, and the meeting package may cost you tens of thousands of dollars, and really this is mainly around the preparation of that package. But as Patrick mentioned earlier, if you've prepared one package, you can leverage that, and it looks very similar across the different jurisdictions. However, if you skip out on this pre-submission meeting and then you go straight to a submission, it can cost you hundreds of thousands of dollars to over $1 million to redo some of the work. Because say, for example, they are not happy with your GLP tox study, that is not only cost, but an extreme hit to the timeline as well. And so do take advantage of the pre-submission meetings, essentially for all of the main jurisdictions that you're interested in pursuing. Engage your regulatory team and or advisors early in establishing the program roadmap and making critical decisions. As you heard from the speakers before me, each jurisdiction has their own timeline. Some are controlled by some legal requirements and others are not. And so having an idea of what those timelines look like should be factored into the program roadmap from the beginning, so that you can kind of work backwards from when you need that feedback to making that meeting request. And then lastly, there is no need to reinvent the wheel here. Leverage people who have done it before and recently, so that if you are interested in going to Japan to do your clinical trial, don't try to figure it out yourself. Do engage someone who's gone to Japan before. That is the end of my talk.
Perfect. Thank you very much, Sam. And before we invite the whole panel up, I just want to say, I think you did a really good job synthesizing some of the lessons learned in regulatory information. So thank you. And I want to fully endorse each of these take-home messages. I think you hit the nail on the head. I want people to consider doing work up front is an investment. It's not a waste of money. Take the opportunity to have pre-submission meetings, meet with multiple regulators. You've heard from 3 different regulators that you may or may not have been familiar with before today, but there's a lot of synergies, and if you can get valuable insights from one that you didn't get from another, it will improve your program. So I think that's a perfect note to end on from our prepared presentations. I think we're doing well time-wise. We have about 25 minutes, I think, left. We plan to do panel discussion and Q&A until 2:45. So if I can invite the speakers, so Omar, Ilona, and Sam, to join me on camera. And we'll go through a few questions that I had prepared beforehand, and I think we'll have some good discussion. And I also see that we've had some questions, the same ones we had at the beginning, that were asked in the channel. So if anybody's attending and has a burning question for any one of or all of our speakers, please do put it down there. I'm looking at it, and Stem Cell Network will help make sure that I get to some of the pressing questions. But maybe we'll start with my first question, and I think we'll start with this will be a question for the regulators, and then we'll get to something more focused from Sam right after. But so Ilona and Omar, I'm curious, what do you hear at Health Canada and EMA respectively about your jurisdiction's priority for cell and gene therapies targeting rare diseases? Do you get some feedback, for example? Does it make sense for developers to prioritize development activities in your jurisdiction more or less? Just really want to know your insights there. And maybe just so this is smoother, Omar, do you mind starting? And then Ilona, do you want to add on if there's anything different in your jurisdiction?
Thanks, Patrick. I think the Health Canada is investing resources in the review of the cell and gene therapies, the areas of rare diseases in particular. This, for example, my division was created. I've put it together 3 years ago, and it's growing. We do have a team internally working on interactions with policy and trying to identify, for example, candidates for the ATP or Schedule G type of drugs. So, and I've covered a number of initiatives and ways to advance these therapies in Canada. That goes to, or that's covered by the modernization of the regulations up to investments in and flexibility that's included in the review process.
Okay, thank you. Yeah, there are some unique programs in Canada that people might be interested in, which could make them a little more interested in Canada as a jurisdiction. But would it be safe to say the true benefit in Canada right now is you are flexible and harmonized. You're willing to listen and have early discussions with groups. Okay, so there may be some things on the horizon, but in the meantime, just the harmonized, flexible approach. Okay, thank you. And Ilona, so I think a number of people are interested just in the jurisdictions they're familiar with and interested in doing their studies in. Are there unique features in European Union people should think of when they decide whether they might start their trials there instead of the US, for example?
Yeah, I mean, also, I haven't gone into the details. We have, particularly for areas of high unmet need, we have specific support programs at the level of EMA, that's towards marketing authorization, and the PRIME program is one of them. I think what we haven't spoken to yet in general is the fact that the development in the rare disease area is usually an abbreviated one. And so personally, I think it's very dangerous to explore lowered requirements and flexibilities early on, because it comes to bite you in the back later on. So from that perspective, Europe starts at maybe a higher level, but you have to eventually meet these standards if you ever want to reach marketing authorization. And if you don't early on, you will run into significant comparability issues, which are really hard to address with ATMPs. And so, yes, we have maybe stricter requirements. We are a large population, obviously, with all the member states that this constitutes. But I also think eventually the sooner you meet the requirements, the better in this setting, because you're dealing with a lot of complexities if you don't. Obviously, I believe in Europe, so I'm biased. So, you have to take that with a pinch of salt. But I think that is a very important discussion to have. And specifically, if you are basically doing a phase 1 pivotal, then it doesn't help you at all to claim flexibility, phase appropriate requirements, but then you want to leverage the same data at marketing authorization. And one thing that I do want to emphasize, because a couple of companies have had issues, the requirements for accelerated approval in the US is a reasonable expectation of a positive benefit risk. The requirements for a conditional approval in Europe, or even one under exceptional circumstances, is a proven positive benefit risk. So it might be because, in Europe, we have health insurance for pretty much everybody, and the health system needs to pay. We need a demonstrated positive risk, not an expected one.
That's interesting. And maybe to close out that thought, so the terminology we use in Canada is that there's the promising benefit, completed safety and promising benefit for a special pathway of a conditional approval. So that is interesting. Thank you. So the high quality standards are a benefit. And I don't think anyone who's thinking from a patient perspective can disagree that it's a good idea, if and when you can, to hit a high quality standard. If you meet it there, then you can take it elsewhere. I think that's an important factor as well. Thank you. And maybe my next question is related, and I'll start with Sam this time, because I think thinking from a company's perspective, what key considerations do you think early stage researchers should consider or assess when planning global regulatory strategies? When you're faced with the opportunity with a very promising treatment, you can go to any one of these jurisdictions. I've already mentioned my biases. I believe a lot of people think of the familiar US FDA and where they think that their investors might want them to conduct studies, and it might be driven by target available market. But outside of that, what do you think some of these researchers should consider in prioritizing where they're going to start and then grow into?
Yeah, I think that's a great question, Patrick. No, I'm not going to order the jurisdictions necessarily because I think that's very nuanced, right? Patient population and all of the other other considerations that we talked about. But Ilona, you raised a really good point, which is around the small population size and how ATMPs can think of like the comparability issue when it comes to this, because you're going to pivot from that early phase stage as a company to late stage really quickly potentially really quickly in terms of a timeline as well. And you don't want your CMC to lag behind. And so thinking about how you're going to think about this program from start to finish is going to help any company out, because no matter which jurisdiction you go to, by the time you're thinking about approval, the requirements are very, very similar. And so it's not about ease of entry, it's about successful completion. And so that first batch that you manufacture and test and release may be your only batch or your non-validation batch. And so think about that as you're thinking about the different jurisdictions. So it's not really about jurisdictional nuances at the early stage, but how are you going to get through the entirety of the clinical trial? Now, I am not naive, and I know that this is a business and there are funding issues as well. And as you hit certain milestones, there's opportunities to raise. So it's very attractive to sometimes thinking about the jurisdiction that you can go into with a non-GMP lot. But what are you going to do after that? And so that's what I would encourage most therapeutics sponsor companies to think about.
If I can briefly add on to that, for me, one of the most salient points in this is also, do you have an adequate product profile? Because nothing is worse than having conducted a hugely expensive clinical trial and then not being able to link that to a defined CMC product profile. Because in my opinion, if you can't, you've wasted all your money.
Wow, and I think that links, I'm going to start mixing together some of my planned questions and some of the questions I'm seeing emerge. And one of them touches on what Sam and Ilona, you both just mentioned, which is, and this is, I think, from Craig, are you seeing in Europe, for example, Ilona, a difference between early phase studies and later phase based on the difference that you noted earlier, that the stricter, higher, full licensed GMP facilities are required in the EU, but not in the US and not in Canada? There's special exemptions for first in human in the US. And then it's down to reviewers who look at this from a product specific perspective when they look at the manufacturing and the manufacturing environment. So are you noticing changes between early studies and later coming from US and Canada and going to Europe? And if so, what's the implication there? If people develop it in GMP-like settings and then have to make manufacturing changes in between early and late studies, maybe some insights into what you might be seeing in these later stage studies, Ilona, would be really interesting.
Yeah, I mean, that's certainly something that's happening, that's clear. But in order to conduct a trial in Europe, you have to comply with the standards. And it's difficult to sort of differentiate because you expect increase of knowledge wherever you started developing your product between early and late phase. The big dangers, and those are cases that we have seen, is that maybe your early batches don't have the same efficacy as your later batches. And those are all issues that are problematic when your overall treated patient population is quite small. And it can happen and it will happen. So it's not that you can pin it down to certain specific issues, I think. But I also think that every regulator takes one look at the dossier and knows whether it's a good one or not. And it's difficult to put it in words why, but you can see it. And certainly certain issues such as report results instead of appropriate specifications are something that immediately raise flags for me. Because even if you don't have manufacturing experience, you should have a scientific understanding of what's reasonable for certain specifications. And this is basically my main point. Developing assays in the lab doesn't compare in cost at all with the cost of a clinical trial. And so for the life of me, I can't understand why you don't put in that investment to de-risk your trial.
Yeah, Sam, do you have something to add to that?
Yeah, and I want to leapfrog off of something Ilona said before. Especially in the rare and ultra rare space, you are potentially using your early phase information for your market application, and so that has to be a reliable product that you've put out there, and that reliability comes from all of the criteria that we've talked about in terms of how you manufacture, test, and release.
Yeah, and just to chip in on that one, the expectation for marketing authorization is that the manufacturing process is representative of the one that's going to be your commercial process. So if you have significant changes between your early data, these data might not be accepted at the level of marketing authorization as pivotal.
Yeah, and having a robust strategy is really important. So people listening should hear this, and that's the main thing: is plan ahead and understand if you might have to make manufacturing changes. I want to bring in Omar too, because I think one thing we should clarify is if you manufacture in Canada or in the US and you want to go to the EU, you can do that. You would have to have a qualified person come in and give the license from the EMA as well. So it's possible to be a Canadian jurisdiction or a manufacturer or a US-based manufacturer, have an EMA registration for meeting GMP requirements in Europe if you're going to conduct studies over there. And so my question for you, Omar, is it helpful when you're reviewing a product that it's already received a third-party review, audit, and things like that from a European qualified person?
Of course, I think the qualified person is our ambassador in a way as a regulator within the manufacturing facility and overseeing what's happening with the quality assurance system and quality systems in general. So that's very, very important and helpful. We're talking about the clinical trial stage. So just to clarify that it's in Canada, we're maybe aligned a little bit more with the US FDA. As Sam mentioned, we do scrutinize the information that's provided in the file, but we do not necessarily flag. It's case by case. Any manufacturer for a GMP inspection at the clinical trial stage. But as you mentioned, having a qualified, a GMP certification or a qualified person, if it's the manufacturer is selling the drug in Europe, is very helpful and important. We do take that into consideration for sure.
Good to know. Thank you. I am going to change gears a little bit. We've been talking a lot about manufacturing and facilities, and I want to switch a bit more to the regulatory and policy side of things. And so my question is, because I hear a lot, people want more guidance in this space. So why don't regulators provide more prescriptive guidance for cell and gene therapies that can, for example, help early stage researchers who aren't as familiar with regulations know more specifically what to provide in a specific indication or a technology space? And Omar, maybe we can start with you, and then we'll ask Ilona and then Sam.
Okay, so first of all, I think gene therapies are highly heterogeneous type of product. So it's very difficult to come up with one size fits all. So it's case by case. That being said, I think there are lots of effort behind the scenes and collaboration among the different regulators to harmonize. We do have ongoing conversation with our colleagues. You have to know that we work with our own different regulations. And as I mentioned, there are efforts to try to harmonize, align. So it's an ongoing effort. Maybe many of you know we do have an ICH working group on cell and gene therapies, and they've met. We do have some work done on that front, and it's again an ongoing effort, and you will have more guidance for specific type of gene therapy or cell therapy products in the near future. But we do align. I think it's coming back to Health Canada, as I mentioned in my presentation. There is alignment in terms of guidance review. Our regulatory framework allows us to use guidance from the EMA or US FDA or other partners. Thanks.
Thank you very much. And I'm going to stay on you for one more second, Omar, because there are two questions specifically directed to you. One is about having guidance specifically for cell and gene therapies. Does Health Canada have one? I think I'm safe to say the only guidance right now is for cell and gene therapy for clinical trials. I'm very familiar with that, having been the lead author on it when I was at Health Canada, but there aren't any others, I think, as of yet, right?
Yeah, not at the moment.
Okay, but you're saying that it is helpful. Someone who has their hands on the US guidance for cell and gene therapy or the European Union can make appropriate references there, and Health Canada can consider that as a rationale for why certain things are done a particular way, and you'll make a science-based case-by-case decision, correct?
Absolutely, and as you mentioned, we think alike. As regulators, it's the same type of mindset. There are small differences, but in general, I think all of us are science-based, evidence-based.
Yeah, and then the second question. Sorry to stay on this, Omar, but I think it's a great question. Someone asked about the orphan drug strategy that was being developed by Health Canada. They noted up until 2022 from Vincent. And I think it's kind of taken a back burner. Maybe prioritization has changed. Can you say what's happening on the rare disease file and policies right now, or if we can expect anything in the near term?
So I think for that, what's being shared in 2022 was a draft strategy with I think the key points, if I remember correctly, a national approach. There was a consultation that went on, and I think there were some principles also shared. For example, equity, sustainability, and evidence-based decisions. And after that, in 2023, Health Canada published a national strategy for drugs for rare diseases, where there was a capital investment. I think it's like $1.5 billion, I think, over 3 years. That was a result of that strategy. But I have to mention, as I presented in my slides, we at Health Canada, we have put in place a number of supportive measures for the drugs for orphan diseases and gene therapies. Like we talked about the pre-submission meeting. I gave you examples of the flexibility. We I also presented I think, two important pathways that are equivalent to other jurisdictions, the priority and the notice of compliance with conditions. And finally, I can elaborate more, but the use of form review, alignment of review with other jurisdictions are important key components, I think, to that are very important for our strategy for these orphan drugs.
Okay, thank you very much. And it's great to have some of the list of things to look into and to know that they fit and they're relevant for this. Ilona, I promised I go to you as well. I do want to know, so rare disease specific guidance in Europe or by member states, are there some things that you want to highlight for people of key resources they might want to look into?
In Europe, we wouldn't have a guidance by single member states. We would always try to have a European guidance. We have just come up with the guideline for investigational ATMPs. I just checked, it's not in Eurolex Volume 10 yet, but it's on the website of EMA. I think the challenge for ATMPs is that you have to consider multiple guidelines, because in terms of quality preclinical, etc., you would have to look at the current requirements through the filter of rare and orphan diseases, and certainly reviewers will do the same. But we always guide by principles. We also, by comparison with FDA, I think we are generally less prescriptive. Interestingly, that feeds back to our legislation. We are running by Roman law, which is case by case and leaves flexibilities. The FDA has always gone very much by precedent in comparison. And I've experienced that myself. The regulators will never take away the responsibility of the developer to consider their own product. That's one thing. And there was another thought I had, which now is running away from me. Might be too late in the day. What was that?
Well, I can give you a minute to, go ahead.
Yeah, and also something I want to touch upon. Europe has never been that good at communication. For the longest time, we've supported the 3 Rs in terms of animal testing. So it will never be the case that you have to do an animal model if it's not relevant. And you might be in a situation where no animal model is relevant, or whether it's only possible to conduct it outside of GLP. And that needs to be justified, would be highly advised to discuss with regulators prior submission, but it is possible. And so conducting an animal study, it doesn't make sense, is certainly not something we would ever ask for.
Thank you, Ilona. And that's for anybody listening, that's a direct reference, I think, to the US FDA's recent in April roadmap to reducing animal testing in preclinical safety studies. It's exciting for a lot of people to see the FDA open up and say, we may not require this, we're phasing it out. There's a 3- and 5-year plan to reduce it. They've been living by the 3 Rs for a long time. And it's good to know European Union is similar. And I think I can say, and Omar, you can correct me if I'm wrong, that Health Canada is similar to the European Union, doesn't have a regulatory requirement to look at studies and would follow ICH S6 R1 and Part 2, which says a relevant animal model for cell and gene therapies is required. And if that relevant model is in vitro, that could be acceptable, but you would look at it on a case-by-case basis, looking at safety and promising efficacy. Is that correct?
Yeah, that's correct.
Sorry, just one minor thing to add, because we've, for the longest time, we didn't really want animal models, but we had them in the proposals, and we would never reject it, because we understand that it was largely done because other jurisdictions required them. So maybe that was a misconception of Europe requiring animal models, but it's not up to us to reject them when somebody does them.
Yeah. Thank you. And we are running up to time. I want to acknowledge there's a number of outstanding questions that I've not gotten to, some of them focusing on N of 1 studies, benefit risk approaches, and then also how to start pre-submission processes. I think that those are great questions that we can help to answer, but we're running out of time, I'm sorry. And I promised the panelists, I want to give them one last opportunity. We'll start with Sam, we'll go to Ilona, and then to Omar. Just to summarize your advice, the final thing that you would like people to take away from something you presented during your presentation or during the panel discussion that you really think somebody should take away if they're planning a rare disease clinical trial in the world about a particular jurisdiction or a key topic. So Sam, if you don't mind starting, what was the key takeaway you'd hope people have from this session?
So I think I was very clear about my first key takeaway, which is have early interactions with the regulators. The second, which I didn't explicitly say, is that I think especially in the rare and ultra rare space, you want to think creatively about how you can achieve what you want to achieve. And so, this kind of touches on the guidance aspect of it. There isn't really a roadmap that you have to follow, but it needs to be scientifically justified. And so do the early work that's necessary to convince yourself and then the regulator that what you're proposing is the right thing to do for the patient population, and then have that early meeting to get confirmation.
Perfect. Thank you, Sam. And Ilona, do you have a key message you want people to take away?
Yeah, think ahead. Getting your trial approved doesn't mean you have the right data for marketing authorization. And don't get too blinded about your own product. So maybe have somebody from the outside take a look at it at some point, because we encounter developers that are slightly too impressed by their own product quite frequently.
Perfect. Thank you, Ilona, for that advice. And Omar, can you wrap us up with some thoughts from what your key takeaway and from a Health Canada perspective?
Yeah, I think it's important to know that we are not lowering the bar in terms of safety and information that's needed to support these drugs. And most of the principles, the regulatory principles outlined in on the ICH guidance documents are applicable in a way to these gene therapies. The focus may be, and again, the clear, I think all of the jurisdictions are welcoming any questions and interactions, like specific risk factors for these gene therapies, like vector related, for example, the off target, in some cases, the cytokine storm syndrome. So I think it's again, it's same principles applicable also to gene therapies. That's what's key.
Thank you very much. And thank you to all of you again for your presentations and your participation in this panel. I know this takes time preparing. I hope and hope everyone will agree. This is very interesting to think about and learn more about Health Canada. We did have the presentation from the Japanese PMDA and from the European Union. So thank you, Ilona. And then Sam, thank you for the perspective, the practical understanding of what we can look at from a client perspective in terms of the sponsors of these trials. We are, I think, about 5 minutes over. We're up to a point now where we have a second session starting in 10 minutes at 3 o'clock. So I encourage everybody to take a short break, come back in about 10 minutes and hear from some of the facilities I think that we have in Canada and for the whole second session. So thank you very much. And I hope everybody has a good afternoon and takes away some of these key messages and has learned something. So take care. And I don't know if Stem Cell Network or National Research Council, NRC, has anything they want to say, or if we should just reconvene in about 10 minutes. Oh, there you are, John.
No, you did a great job, Patrick. Yes, we'll be back in 10 minutes. We'll just go into a short break and then we'll open up. So please don't go away. Go refresh and be ready for more good stuff. Okay, thank you, everybody. Okay, I think we're getting close to the top of the hour.
I think so.
So welcome back. I hope you've all been able to top up your coffees or refresh yourselves. And now we're going to start session 2, which is cGMP gene therapy manufacturing capabilities in Canada. For this session, I'd like to introduce Dr. Kelly Parato, Director of Research and Development at the Human Health Therapeutics Research Centre at the National Research Council of Canada. In her role at the NRC, Kelly fosters multi-sectorial research collaborations to support the translation of scientific health research into innovative medicines. Kelly, over to you. Thank you very much.
Great. Thanks so much, John. And thank you to my NRC and our colleagues over at Stem Cell Network for organizing this great workshop this afternoon. I think it's very interesting and very timely. So I'm delighted to be here to moderate this panel session today. As John mentioned, currently I'm the Director of R&D for Bioprocess Engineering here at the Human Health Therapeutics Research Center at the NRC. And here at the NRC, we have expertise in this department and across the organization that includes sort of expertise and specialized facilities to work with collaborators and clients and companies, Canadian and abroad, to essentially move their biotherapeutic or vaccine platforms and products from the discovery and lead selection stage through process development and into pilot scale up R&D. We've been doing this in R&D for the last couple of decades, and we've worked with dozens, if not hundreds of companies and made some substantial gains in helping to advance value creation for some important products in the marketplace. And so the more recent GMP capabilities that we have that you'll hear about later today is a more recent addition to our value chain. So now we can extend those offerings in product development now into sort of the phase 1-2 clinical trial space. And this came to us as a federal investment as part of ICED's biomanufacturing and life sciences strategy, sort of during and post-pandemic. But there are, of course, several very significant biomanufacturing facilities and capabilities already existing or newly grown in the Canadian ecosystem, whether these are industry-founded or academic or nonprofit. And together, these organizations collectively offer technology developers very robust capabilities to accelerate clinical or commercial development of Canadian innovation. And these different facilities and organizations have a variety of capabilities to support development of varied production modes, platforms, and product types. So this session really is aimed at demonstrating across to the symposium audience the various capabilities that exist in Canada. And our presenters will specifically highlight how they have capacity to support therapeutic development for rare diseases more specifically. And so this session will give you an up-to-date overview of Canada's GMP manufacturing landscape. We'll highlight the current capabilities and perhaps also future growth potential. And in this way, by mapping the available resources and expertise that exist, this session is aimed to help stakeholders navigate sort of manufacturing challenges that align with with product development programs. And will also help this stakeholder audience to identify opportunities to access resources through collaboration and otherwise, and to understand how Canadian infrastructure aligns with international quality and regulatory standards. So we have in this session 6 different flash talks of 5 minutes each. We're trying very rigorously to keep to time, and we'll signal in the chat as you're approaching the end of your presentation time. And we'll be keeping questions like in panel 1, we'll keep the questions for the Q&A session towards the end. So what I'll do is I'll just I'll introduce all of the speakers all at once, and then one by one, when it's their turn, they'll turn their camera on. So first, we're going to have a talk from Jessica Madigan from BioVectra. Then we'll hear from Minh Tran, the Director of the Clinical Trial Material Facility here at the NRC. Third, we'll hear from Jen Queasy, the Director of the Biotherapeutics Manufacturing Center, the Virus Manufacturing Facility at the OHRI. Number 4 spot, we'll hear from Jessica Wilson from Canada's Michael Smith Genome Sciences Center. Number 5, we'll hear from Denis Claude Roy, the Chief Scientific Officer of C3I. And finally, number 6, Jessica Tate, we will hear from CCRM, the Center for Commercialization of Regenerative Medicine. So with that, I'd like to welcome Jessica Madigan to turn on her camera from BioVectra and get us started.
Thank you. Thank you to the organizers for inviting me to speak today, and thank you to the attendees for joining. I'm Jessica Madigan, the Director of Business Development for Nucleic Acid Modalities at BioVectra. With innovation as our main driver, BioVectra is taking steps to partner with our customers to bring better medicines to more patients. Since 1970, we have been serving our global clients by providing superior quality and expertise as a full-service CDMO for biologics and small molecules. Our core driver over the past 5 decades has remained the same. We care about making better therapeutics. Headquartered in Charlottetown, Prince Edward Island, we have 8 facilities total between 2 provinces, Nova Scotia and PEI. Our 5 distinct GMP manufacturing facilities all operate under a single mature quality system that is compliant with ICH Q7 and Q10. We supply raw materials, APIs, drug substances, and drug products for all phases of drug development, including preclinical, clinical, and commercial supply. We have a strong record of successful inspections by regulatory bodies, including the US FDA, Health Canada, and Japanese PMDA. In an effort to address the need for in-country manufacturing options for future pandemic responses, in November of 2021, the Canadian government granted BioVectra necessary funding to build a facility to manufacture life-saving mRNA vaccines. The new state-of-the-art facility is now completed and GMP operational. This facility is located in Prince Edward Island and offers clinical grade to commercial grade manufacturing, end-to-end, including plasmid, messenger RNA, LNP, and fill finish. As you can see on the right-hand side of the screen, with respect to cell and gene therapy products, we support plasmid DNA production. High-quality plasmid DNA is a key component in cell and gene therapy manufacturing, with applications including templates for mRNA production, production of AAV and lentiviral vectors, and pDNA is also used to deliver genomic material used to express chimeric antigen receptors for CAR T cell therapies. BioVectra has an optimized platform process for manufacturing high-quality plasmids. Our platform includes optimized clone selection strategies, high production efficiency and fermentation, specialized continuous lysis systems, and flexible chromatography methods. By using a platform process that has already been tested against a large variety of plasmids, you can be highly confident in moving quickly from project conception to the product release. We also offer fill finish and final product. Your product is unique, but your aseptic filling needs are not. BioVectra has 2 VanRx filling lines for faster turnaround time, and some key advantages to the VanRx equipment include single-use flow paths and automated filling for low risk of failure, ready-to-use containers and closures, which eliminate the need for vial prepping, press-fit closures, which are applied all at once, which lowers the risk of contaminating particulates. And the use of this equipment can decrease project timelines, as BioVectra has already validated the fill process for 10R and 2R vial sizes. We also offer quality control and release testing. So along with our manufacturing solutions, we do aim to control the timelines and the quality of our products by developing, qualifying, and validating analytical methods in-house. Each modality has specialized methods adapted to characterize and quantitate purity, identity, process-related impurities, and the safety profile. And finally, we offer supply chain and logistics. Next slide, please. In addition to supporting these services, we have a wide range of competencies. We have some of North America's largest microbial fermentation capacity with 2 by 17,000 liter fermentation vessels in our commercial biologics facility. In addition to our competencies in microbial fermentation, we offer synthesis of lipids, MPEGs, linkers, peptides, and highly potent APIs. We also have a catalog of high quality and GMP grade bioreagents, including DTT and TSAP. And our nucleic acid services include plasmid, messenger RNA, LNP, and fill finish services. In other recent exciting news, BioVectra was acquired by Agilent Technologies. With this acquisition, BioVectra's capabilities in biologics is being combined with Agilent's expertise in oligonucleotides. Specifically, we're looking at advancing world-class capabilities to support gene editing. Agilent and BioVectra can provide customers with a single source for gene editing technologies, including guide RNA, plasmid, mRNA, LNP, gene editing proteins, RNP complexing, and fill finish. We are advancing our unique technologies which leverage our platform and customized manufacturing solutions and are designed to overcome supply chain complexities, capacity constraints, and scalability challenges, which are key limitations to success. So I invite you to contact me with any questions, and I look forward to supporting the advancement of Canadian cell and gene therapies and rare diseases. Thank you.
That's great. Thanks so much, Jessica. Next, we have Minh Tran from the NRC.
Can you see me?
We see something in your camera. I don't think we're seeing your face though.
How about this? Is it better?
There you go. Perfect.
Okay, great. Sorry. All right. So thank you very much, everyone. So just a little bit of a background of CTMF here at the HHT. So we're actually headquarters in Ottawa. Myself and Kelly, we work very hand in hand in regard to the value chain. The actual site, it is located in Montreal facility at Royalmount. In regards to our GMP capability, so when we look at it, we have up to about a 500 liters bioreactor capacity between from a smallest size to about 5 liter all the way up to 500 liters. And we, like Kelly mentioned, we cover from phase 1 and 2 only. The actual process capability we have here at CTMF is really to perform upstream and downstream manufacturing only. So anything that regards to formulation, fill finish, secondary packaging, and QC testing for release. One of the, I guess, our capabilities is really the partnership that we do with all the industry, either with BioVectra or any other organization in the Canadian ecosystem. In regard to the technology platform, so what we use is most biopharmaceutical companies, we use single use platforms. So all our manufacturing bioreactors or mixers use single bags. So that's to spread really cross contamination and also in regards to cleaning time. In regards to our process capabilities, we can manufacture mammalian and insect cells specifically, and also in regards to biovector. So there's only 4 types that we are able to manufacture: AAV, adeno, retro, and lenti. In regard to our capabilities, we are just a service organization. We do a little bit of support, but we don't do any type of development as we are a cGMP manufacturing facility. One of the things we do accommodate also is manufacture a master cell bank. So that's one of the actual capabilities that's been added to our facility. A lot of the, I guess, clientele that comes to us are small and medium enterprise, but really from a clinical stages, right? Anything that relates to process development, that's kind of like more where Kelly's department is from a bioengineering. So the way the NRC or HT works is really client can come in from the early stage, from an idea, from Kelly's side all the way upstream. Where we are is really the GMP side, which is the end downstream of the process. And we make sure that the product is manufactured based on cGMP regulatory requirements. And the 2 most important things that our facility does accommodate is the GMP compliant to Health Canada specifically, but also US FDA. So when the facility is built to accommodate both of those agencies. Next slide, please. So I just want to talk a little bit of the key challenges in regard to the gene therapy specifically. The first challenge that we get to see here is really from a clinical trial and commercialization facility here in Canada. I come from an industry where the Canadian ecosystem, when it comes to cell and gene, it's still very small, and it's actually growing since the pandemic. One of the resolutions that our federal government has invested a lot on is really the construction's investment of a $2.5 billion, the construction of the NRC CTMF here at Royalmount, and then also the commercial size facility that's also annexed to the NRC Royalmount is our BMC facility. In addition to this type of investment, OmniBio was also built and also invested by the federal government to really help grow the cell and gene landscape in Canada. The second challenge that I see from a Canadian ecosystem is really the talent pool. As we know, biologics is a growing industry, but the talent pool, I believe that we're still growing as we go along, especially in cell and gene. So one of the resolutions that I kind of like seen with the University of Guelph is the creation of the Canadian Advanced Therapy Institute, which also links to the OmniBio folks. So I think it's very important that it's one thing where we can grow the ecosystem, but also the talent pools is very important to continuously bring the expertise, but to ensure that the expertise is maintained for future product and future innovation. The third challenge, what I see in regard to the gene therapy specifically, is really the cost. I'm sure, as we all know, I think right now there's about 4 specific products that are approved by Health Canada, and only about 2 of them are pretty much subsidized by our provinces. As we know, each of these products specifically cost above $1 million. And again, we know it's a rare disease. It's not every Canadian that has it, but also to access these medications, it's proven to be a huge challenge. So one of the things is, I think through the Canadian Drug Agency and through a lot of these nonprofit organizations, the Cancer Research Organization, and some of our provincial organizations, where we're trying to push as hard as we can for reimbursement strategies so that we can help kind of open these accessibility of these products to as much as Canadian as possible. But again, I think that's challenge number 3. It's still an ongoing challenge for the industry, but also from a patient standpoint, where it's still very limited, this access to these medication. Right, thank you.
Great, Min. Thanks very much. Next, I'd like to welcome Jen Queasy from the OHRI.
Hi, everyone. Thank you so much. As I mentioned, my name is Jennifer Queasy. I'm the director of the Biotherapeutics Manufacturing Center here in Ottawa, also known as the BMC. Could you go to the next slide, please? So very briefly, we are located at the general campus of the Ottawa Hospital, and we have been manufacturing therapeutic cell and virus products for more than 17 years that have been used in early phase clinical trials, both here in Canada, the US, Asia, and in Europe. The way we do this is we employ a variety of different platforms within our facilities. That includes scalable bioreactors, both supporting adherent cell culture as well as suspension. For adherent, we're up to 170,000 square centimeters using bioreactors like the Roller Cell 40. And from a suspension perspective, we go up to 200 liters currently. Of course, depending on the indication in which the therapies we're helping support manufacture are going to be tested in, we offer a variety of different purification strategies, usually focusing on a combination of TFF and chromatography. Oh, sorry, went back. Thank you. Our organization is unique in that we are a not-for-profit contract development and manufacturing organization, and we do support end-to-end services for your products. So for instance, we can go so far back as helping you engineer your virus of interest, in the case of vectored products, all the way through to leveraging our existing platforms for vector and cell therapy manufacturing on through to GMP compliant manufacturing and including as well GMP QC release testing of your final product. I also would like to highlight what's unique in working within a hospital environment is that we do have very functional and tangible links to the hospital infrastructure by way of implementation of clinical trials that result from the products that we manufacture. It's not to say that there is a requirement to use the Ottawa Hospital to do your clinical trials, but again, this is something else that we offer by way of a network. If you're looking for somewhere within Canada, in Ottawa in particular, to run your clinical trial. And as a nod to that, we also do support regulatory submissions. So by way of helping write chemistry, manufacturing, and control sections of your CTA application. I want to also mention, actually, if you can switch to the next slide, as I mentioned, we do, there we are, thank you so much. We make both cell and virus products, and in some cases, we do a combination of the two. So a perfect example would be as far as our lentiviral platform that we have that has supported the manufacturing of lentiviruses for our wholly made in Canada CD19 CAR T trial. And also want to say that as of the end of this year, we'll have the capacity to also make the CAR T cells themselves. So both the vector and the cell product that be used for that CAR T cell therapy we will have the capacity to do that here at the Ottawa Biotherapeutics Manufacturing Center. We have dedicated facilities to support all the activities I already described to you. So dedicated process development and assay development suite. We have six independent GMP clean rooms of various different grades to support both the cell and vectored programs, more than 40 staff. And I would say something that's very unique to us as well is that we are home to Canada's only work integrated learning program in biomanufacturing, where we're helping to support the training of the next generation of highly qualified personnel who will be participating in this space, hopefully when they graduate either their college or university programs, which has been a really tremendous feeder, both for our facility from finding skilled FTE, but also to help seed the ecosystem. The last thing I'd like to mention is that our facility has been supported by a variety of different funding bodies, including the Canada Foundation for Innovation, BioCanRx, the Ontario Research Fund as well. And recently we were awarded significant funding, $70 million from a combination of federal and provincial funds to build a brand new facility, which will more than double our current capacity, allowing us to bring on additional processes and product lines, which will be operationalized by March of 2029. So thank you so much. And I will be around obviously for the panel if there's any questions.
Fantastic. Thanks so much, Jen. Next up, we have Jessica Wilson from the BC Cancer Research Institute.
Great. Thanks for having us and letting us talk about our services. So our facility is located at the BC Cancer Research Institute in Vancouver, and we specialize in the production of plasmid DNA at scales ranging from milligrams to ambitiously grams, following phase-appropriate GMP guidelines. These plasmids are critical components used to manufacture mRNA and viral vectors that support early phase trials here in Canada. Could I get the next slide, please? Yeah, great. Our plasmid manufacturing facility was first established in 2018 in the research lab of Dr. Robert Holt, which is part of the larger BC Cancer Research Immunotherapy Program with support from BioCanRx and the BC Cancer Foundation. We were able to get the facility up and running quickly thanks to the Holt Lab's extensive experience in vectorology and plasmid production, as well as a comprehensive quality management system that was already in place for the clinical genetic testing at our Genome Sciences Center. Our immediate goal was to produce the plasma needed for CLICO-1, which was and is Canada's first investigator-led CD19 CAR-T clinical trial. CLIC, or Canadian-led Immunotherapies in Cancer, is a Canada-wide program that aims to bring together researchers, clinicians, and patients from across the country to make access to these innovative treatments more affordable and accessible. CLICO-1 has now seen over 90 patients in BC and Ontario receive treatment so far. So at the time, around 2016 to 2018, there were no Canadian suppliers for the amount and quality of plasma DNA needed to support an early phase trial. While commercial suppliers have since emerged, our focus remains on producing plasmid for academic scale, high quality or phase appropriate GMP plasmids that enable these investigator-led trials. Following the success of CLICO-1, we've leveraged our initial investment and substantial effort in setting up the plasmid manufacturing facility to support additional projects. We've manufactured the plasmids for CLICO-2, which is the second CLIC trial for a CD22 CAR-T cell therapy product, which opened earlier this year, as well as collaborating on several other small investigator-led trials with other Canadian institutions, including an N=1 trial. Our operations work on a cost recovery basis through service agreements with our collaborators. So we're fulfilling a not-for-profit need that is attractive, especially to these academic funded projects. We can work with plasmids provided by our partners or with plasmids that leverage our clinically tested proprietary lentiviral vector system. Our manufacturing platform covers the entire plasmid DNA process from bacterial cell banking and culture through harvest, purification, and QC testing. Every batch is issued with a comprehensive certificate of analysis, and we maintain a robust QMS with SOPs that control every aspect of manufacturing, testing, and facility operation. Next slide, please. Over the past year or two, we've expanded our capacity by optimizing our bacterial culture and purification pipelines. These improvements allow us to boost our output quite dramatically. For some plasmids, we've seen up to a 6-fold increase in yield. We've also expanded our quality testing portfolio to meet the evolving regulatory landscape by adding new validated assays for measuring residual host RNA and DNA, as well as supercoiled isoform content. Finally, we've applied our expertise to solve some more complex technical challenges. For example, AAV plasmid production, we encountered issues related to inverted terminal repeats, ITRs, which can make the plasmids unstable. Through our custom sequencing strategies and assay redesign, we're able to confirm sequence integrity and refine our release testing methods. In short, the plasmid manufacturing facility here at the BC Cancer Research Institute has grown from a single project to a national resource, enabling Canadian academic investigators to launch innovative cell and gene therapy trials. We remain committed to advancing this mission by continuing to improve our processes, expand our collaborations, and provide the high-quality plasmids that are essential to Canada's clinical research ecosystem. Thank you.
That was great, Jessica. Thanks so much. Next up, I'd like to welcome Denny Claudois from C3I.
Thank you very much, Kelly. Do you hear me?
Yes, we do.
Okay, great. Thanks very much for the opportunity to present. I'm a professor of medicine at the University of Montreal, also a CSO at the C3I Center. So if I can have the next slide. C3I is based in Montreal. Yeah, thank you. So it's actually based in Montreal, but we are linking with centers throughout Canada, and actually have formed a network, and actually will continue that through CellCan, which is a Canadian regenerative medicine and cell therapy network that was built out of the stem cell network. And C3I is actually another offspring, which is a CDMO that's oriented towards cell and gene therapy. So it's the facility is a 14 clean room environment that's actually GMP certified, which is unique in Canada, and it's at the CTC, which stands for Center of Excellence for Cell Therapy, and it's at Maisonneuve-Rosemont Hospital. C3I is a for-profit organization that has been performing phase 1 to phase 3, as well as commercial stage for advanced therapeutics medicinal products. So actually cell therapy and gene-modified cell products, and also been generating recently some gene vector facility product. So what is interesting is we really want to start working with the researchers, help them develop their product, and bring them into the phase 1 to phase 3 area, and even commercial scale, which is, and we have right now a product that has reached this level, which is a UM171 here in Montreal, and has involved people from all across the country. So this is very exciting. So in terms of platforms, we have suspension, bioreactors, automation, open and closed systems, Clinimax, Prodigy, cocoons, and the people I mentioned, it's really from early stage and even before people do the clinical development, to help them develop so that rapidly you can jump into being able to do a clinical trial. In terms of type of therapy, we've been doing a lot of ex vivo modified cells, so genetically modified cells, in vivo administration of vector. That's a very early endeavor with autologous as well as allogeneic cells. The ex vivo cells are administered to patients. In terms of unique value proposition, I think we're the first GMP certified center certified by Health Canada, and we're certified by the EMA. We just learned how extensive both organizations are, have high regulatory standards. We actually have a DEL, which is a drug establishment license. We have personnel that's been working for more than 20 years in the area of R&D for cell and gene therapy. There's more than 15 years of experience in GMP manufacturing. 400 innovative products have been given, and what is kind of unique is that we have above 95% success rate and currently a 99% success rate in the production. And we do gene modification, CRISPR editing, viral, non-viral transduction, and AAV production. And to come, we're right now in the middle of construction for modifying our unit to add an lentivirus in large scale or medium scale, I should say, AAV production. So we're able to help with vector design and engineering. We have viral vector manufacturing right now, but clearly it will expand significantly over the coming 2 years. Cell processing and genetic modification, we do small scale fill and finish. We have quality control and release testing, and we do have a supply chain and logistics, which is integrated. Next slide. So actually what's interesting is we're able to help everybody produce, like in terms of GMP manufacturing, but we also have laboratory services that provide all the development, the validation, but most importantly, the quality control for all the different testing, including specialized clinical testing. And then we have a clinical research environment and unit that helps support the development of the cell and gene therapy. So it's actually a one-stop shop where we provide all these various services. And you have on the left a list of the type of work that is being done either in cell culture with stem cells, immune cells, and other tissues. We've actually been producing these various cells not only for Canada, but Canada, United States, and Europe. We've been shipping cells regularly to the US. Even with the current environment, I think Canada remains an interesting area for cell manufacturing. And I'm sure that we're hearing today that there's many opportunities to continue in that area. And the type of work that we do, you can see here, we can enrich cells, we can do immune selection, we can activate and expand. We do gene modification, CRISPR editing, viral and non-viral transduction. And we have a closed system AAV production unit, and as I mentioned earlier, developing the full GMP lentivirus and AAV production unit. So hopefully we can help researchers from all across Canada to help them bring their products to the patient, and that's why you did that. So back to you, Kelly.
Great. Thank you so much, Denny Claude. Okay, and last but not least, Jessica Tate is here from CCRM.
Sorry, it's taken a minute for my computer to catch up. So thank you. Yes, I'm the VP for Vector Technologies, technically at CCRM, but you'll hear me speaking about Omnia Bio, and I will discuss the relationship between the two organizations. And just a quick fact about myself, I've been in the gene therapy space for about 20 years, particularly on viral vectors, and in the CDMO setting for the last 11 years approximately. So to fully appreciate the expertise at Omnia Bio, which is listed here on this page, I think it's important to understand how CCRM's background and how that ties into Omnia Bio. So about 15 years ago, CCRM was founded as a nonprofit organization to leverage Toronto's world-leading stem cell capabilities as well as other hGMP technologies. 5 years after that, as we entered in a partnership with Cytiva, and it was under that partnership where we started to get more into immune cell therapy, so T cells, NK, B cells, things like that. and as well as vector manufacturing. In 2019 is when we moved into providing a CDMO service. So we opened our phase 1, phase 2 manufacturing facilities at our CCRM site in Toronto. And that those services grew to the point that it justified us spinning out as a new entity, which is Omnia Bio. Omnia Bio is now our sole CDMO business. So we moved the CDMO function out from underneath CCRM and put it underneath Omnia Bio. Omnia came into existence around 2022, I believe. And then last year, we had our grand opening of our flagship commercial facilities. That between the Toronto facility and the Hamilton facility, we now have about 160,000 square foot of GMP space that includes 26 different clean rooms, 20,000 square foot of process development space, and 10,000 square feet of QC space. The Hamilton site is, again, more for commercial product. We can do early phase to pivotal at our Toronto site, and then our Hamilton goes up to commercial, and we recently acquired our Dell as well. Through this journey, you can imagine we've developed extensive capabilities in both allogeneic and autologous cell therapies. Those cell therapies include gene-modified cells. The genetic modifications can occur through gene editing or vector-based gene delivery, and that's both viral and non-viral vectors. We do have licenses to gene editing technology, and we have the manufacturing capabilities, again, of various vector types, viral and non-viral. For a full list of the cell lines that we've worked on, as well as the vector capabilities, you can visit our page or reach out to me, and I'm happy to provide that. The vector grades that we make are suitable for ex vivo as well as in vivo. And all of these manufacturing platforms have in-house state-of-the-art analytics. There's only a couple of assays we still outsource, mostly around the safety testing. And then finally, we do have access to pre-banked GMP grade iPSC cell lines through our subsidiary Linea Bio. So really the only capabilities that we're lacking in-house is for vector design and plasmid manufacturing, but through our ecosystem, as you've heard with previous speakers, we have Canadian partnerships to provide that service, and we're happy to manage that through subcontracting, or our customers can go directly to those organizations. And then lastly, our main goal, I think, as an organization, or how we want to position ourselves, is partner first. So we don't see our customers necessarily as customers. We see them, again, as collaborators. As such, we try to be flexible and have customized solutions for our partners. So even though we have collaborations with Cytiva, we are not we're agnostic for technology. So any platforms that customers are interested that we already have, or we can bring in site if they so desire. And then, again, we can service from preclinical IND enabling and then to commercial. With that, I'm just checking to see if I missed anything. I don't believe so. So thank you, everyone, for your time, and I hope you all reach out to me or others at Omnia Bio in the future.
Great, thanks so much, Jessica. So I think we have a short Q&A session. I'd like to invite all of the panel 2 speakers to put your cameras back on. We can certainly go through any questions. I first wanted to start by congratulating everyone for giving such a great comprehensive overview of each facility in 5 minutes and really sticking to time. But I think these were all really interesting, and I can see where there's a lot of synergy and a lot of complementarity in what's available to support the rare disease space in Canada. So I'm just turning it over. I guess I'm to look in the Q&A. If there are questions that have come from the audience, please go ahead and place them there. We have one question: is there any siRNA and mRNA manufacturing facility? So I don't know that came up in the presentations with this group, whether there are current capabilities for siRNA or mRNA, although perhaps BioVectra, and Jessica's got her hand up.
Yeah. I can answer that. We at BioVectra have messenger RNA capabilities in-house in our Canadian facilities. Agilent has recently acquired BioVectra, and they specialize in siRNA. Their facilities are located in Colorado, and they're one of the premier manufacturers of siRNA globally. So, through our network, we can, of course, introduce you to the appropriate contacts there.
I'd also like to
Great.
add, we don't have the capabilities right now. So to your point, Kelly, about new capabilities, we are working with a partner right now to actually develop out a platform, and we would anticipate that this would be one of the new product offerings that we'd be having in our new facility in 2029.
Yeah, great, fantastic. So I don't currently see other questions yet from the audience, but I certainly have a few myself. And actually, Jen, picking up on your comment, is there anything going around the table that anyone wanted to highlight as emerging capabilities that they're expecting in the short-term horizon that they didn't get a chance to mention during their presentations? If anyone wants to raise a hand from the panel. Jessica Tate.
Yeah, so I think one of the things we were seeing and we're trying to get ahead of right now is some of these more innovative in vivo vector design. So whether it's what I'm calling protein wagons or lipid wrapped virus-like particles, but we're seeing a lot of really interesting novel ideas of what I like to call Frankenvectors, but piecemeal of different types of vectors. And so we're trying to get our manufacturing and analytics ahead of that.
Yep, great. Jessica Madigan.
I didn't mention it in my previous capabilities, so I want to mention it here now. We've recently added master cell banking capabilities at BioVectra. That was one of the biggest bottlenecks for us to be able to manufacture plasmids, was outsourcing master cell banking and having the cell banks tested before going into GMP manufacturing. So we now have that in-house, and we can decrease those turnaround times.
Great. And Jen PZ.
Yes, so in addition to the vectored products I had listed on our slide, also want to mention that this year, for the first time ever, we manufactured a virus-like vesicle, and we'll be manufacturing exosomes towards the end of this year and into next. So that's a very exciting new platform, kind of on the vectored side, if you will. On the cell side, as I mentioned, we will be manufacturing CAR T cells, and that is obviously with a lot of assistance and support from our colleagues out in Victoria, BC, but also we'll be bringing online the capacity to manufacture tumor infiltrating lymphocytes as well. So TIL products. So very excited about the potential there.
Oh, very interesting. Denny Claude.
Thanks, Kelly. Just wanted to add that we're collaborating closely with people that are developing various viral vectors. They're vVector Bio, which is actually located at McGill University in Montreal. There's also at Université Laval. There's also the Cerveau group, which are actually working on developing a number of new vectors. So actually, we're well connected with these people, which I'm sure they would be very happy to connect with all the researchers in Canada and companies, startups that are interested in developing these further.
Great. Thank you. I wanted to, I had another question. I was sort of interested because there's a variety of different flavors of organization here with different capabilities and who work in predominantly, industry or academic sector. I'm curious to know what each of you think is sort of, what's the biggest challenge or barrier to working with the Canadian pipeline when a new collaborator, new client comes in your door? What are some of the common gaps that you see that stand in the way of getting off to the races quickly to execute a project? And are they more technological, or are they more about funding or regulatory? What has your experience been? I guess particularly where we're talking about the rare disease space, I should say that. Jen, go ahead.
So this wasn't planned, but to kind of speak to, I guess, the first session, I think a lot of times we have folks from a variety of different backgrounds, whether they're academics doing this for the first time, our startup companies or even larger companies that maybe don't have a sense of exactly what they need us to do. So I do want to make a bit of a pitch for some of our speakers from the first session to say, engaging with regulatory consultants, getting that expertise so that you can come and direct a group like mine and the other folks here on the phone as to exactly what you're looking for, helping us build out your specification, I think is really worth its weight in gold. I loved Sam's comparator there. 10,000 up front is nothing to $100,000 later if you have to redo things. And very much when you start engaging with us in the manufacturing space, that is the magnitude of cost you're kind of starting to look at there. So I would say that's the big one. And then the second one, which I'm sure everyone here has experienced, supply chain is a problem. It is difficult. And even now, when it comes to trying to plan out GMP runs, it's really dependent on our ability to get the materials in a timely fashion. So, obviously a call to action maybe for folks here, if there's some way we can combine our resources such that we can make sure that Canada is well represented when it comes to getting the materials we need to provide this very important service, I think that would be very helpful.
Yeah. And Danny Claude, do you have a thought?
Yeah, well, I think that the big problem, or one of the big problems, is that people are coming late to try to discuss with the people that are going to manufacture. Like, they've done a lot of experiments, they've done great science, but they haven't put a lot of as much thinking as to how are they're going to apply this to the clinic. And you have to go back to the bench to do additional experiments. And if you really want to save time, you really need to connect with the manufacturer very early so that you can already build in this mentality into your experiments as you're developing your product. And I think this is key. And too often people think about the science, which is great, which is definitely needed, and that's what we're strong at, in particular in Canada. But we have to be strong everywhere. And you have to be thinking about that very early. And maybe one way of getting people to understand why, how this works, is to get involved with organizations such as CATI, that is doing training and has a facility in Guelph that was mentioned in the presentations. Kathy is training people for very reasonable cost, both from academia and from industry. So actually, we have quite the chance to have this organization there.
Great. Thanks, Danny Claude. And Jessica, you wanted to add?
Yeah, and I think it's kind of echoes statements others have made, but maybe in a different way. It's don't focus just on speed and low cost up front, right? I mean, I think you see a lot of CDMOs. I know previous CDMOs I've worked at have advertised, oh, I'm going to get you your first GMP batch within 9 months. Well, that looks great on paper, but how many corners are you cutting? Or what pitfalls aren't you prepared for? What data aren't you going to get that you might really wish you had later on? So that's just my one advice that I always give people is, don't just focus on the dollar sign and the timeline, like really have those in-depth conversations with your manufacturer.
Yeah, that's great advice. So we're a little over time, but I know we have some minutes left before the closing remarks. I'm going to ask one question that came from the Stem Cell Network chat, and then colleagues moderating this session can let me know if we have time for more questions, because I see a few others. But a question that comes is, across the facilities, what is your expertise or related financing supports that are available for cell or gene therapies that are coming out of academia or early stage biotech, since money is always an issue? So this might touch on, collaborative pricing models or co-development of proposals to get funding from various sources. Are any of you engaged in these ways and have any kind of solutions to bring to the table to address cost? Jessica.
Just speaking from CCRM's perspective, so CCRM, it has they want to get their hands and help in any way that they can. And some of the ways that they have that is through our Medicine by Design, which works with academic partners to help them better design therapies for use. We have our Enterprise Foundation, which is an investment. So that's where we'll award pretty early, relatively small dollar sign kind of investments for early startups. And then as well as we have our company creation arm, where we help customers take technology from academia and turn it into a commercial product.
Okay, great.
And
Did anyone else want to chime? Oh, Denny Claude?
yeah, just add very quickly at C3I, what we have developed is actually a strategy to help people, to help them identify the right sources of funding and accompany them through the development. So I think that's something that we found was necessary for a lot of people to help them look at what's the lay of the land and what are the opportunities. So C3I has developed a special interest in accompanying people to get the appropriate funding, which is not easy, as you indicated, Kelly. This is a significant hurdle, as these all of these approaches are pretty costly, unfortunately.
Yeah, no, absolutely.
Can I
Okay.
one thing?
Sorry, go.
Just
Go
want to say, that's why I think it's so important that we do have funded networks like Stem Cell, like BioConnectX, that recognizes that this is an unmet need as far as funding, because to the person's question here, absolutely, there's a massive gap when it comes to funding. We can't necessarily rely on the government of Canada to provide all the funds, but at the same time, at the stage that they're engaging us at, oftentimes it's not de-risked enough for private capital to be stepping in. So it's definitely a gap, and it's expensive one at that, because again, considering we're manufacturing things that are going into people, you know, the cost is just that much more because the criticality of the product that's ultimately being made here.
Yeah.
Yeah,
No,
Stem Cell
absolutely.
Network. Oh, sorry.
Go on, Denny Claude.
Yeah, just Stem Cell Network, indeed, as Jennifer indicated, has been a key leverage asset for us Canadians, actually, in all the area of regenerative medicine and stem cell transplantation. There are organizations in the area of cancer that are also Cancer Research Society, the CIHR, and others. So actually, there are a number of organizations that are really helping in their opportunities there.
Yeah, absolutely. We do have more questions in the chat. I'm told I have 3 more minutes for questions before we'll close. So one of the questions, any capabilities on GMP biorepositories for clients' GMP master cell banks? So actually storing the client banks. Does anyone have any leads or capacities for that? Jessica Madigan.
We have capacity to store microbial cell banks. So it just really depends on, you know, what type of cells they are. But at BioVectra, we store microbial.
Okay.
Yeah. We have capacity at C3I to store cells in terms of master. We do have master cell banks that are stored, but this would have to be discussed, obviously, depending on what's the need, what's the size, and how can we help. So it needs to be looked at.
And similarly, same thing with Omnia Bio, and we partner with an external storage as well, so we can de-risk by, you know, so it's not all in one site. So yes, we can facilitate that, but it does involve a discussion.
Okay, great. And we might have time for one last one. So compared to a few years ago, we now have several facilities in Canada for biomanufacturing, which is great. Do you believe there's enough trials and demand in Canada to keep everyone busy? So speaking to the robustness of the pipeline.
I guess I'll kick it off.
Sure, why not? Your microphone's on first. So
well, fortunately, unfortunately, I'm not sure. But to date, a lot of our customers have been from the US. So I don't know if we can say there's enough clinical trials or not, but for whatever reason, a lot of the pharma and biotech companies that have been deploying our CDMO services have been from the States.
Okay, interesting.
From BioVectra's perspective, I would say like sustaining on Canadian business alone is probably not going to work. But you know, we serve customers globally. We work in all different regions of the world, and of course, you know, we love working with our Canadian partners. But yeah, we work in a global footprint.
I just want to echo both of BioVectra and C3M's responses there from a BMC perspective, but maybe kind of put a little aspirational thought out there. I think that Canada's veritable lack of biomanufacturing capacity historically has kind of led us to the point where we don't have as much biotech in Canada. So I'm hopeful that with the additional biomanufacturing capacity we will have here, we will be able to anchor these new startups, attract larger pharma companies to wanting to do business in Canada. And who knows, maybe, you know, it flips in the other direction in the next 10 years where we're predominantly working with Canadian businesses because we've been helping basically give them the opportunity to stay here and grow.
Yeah. And in order to do that, we need to encourage our scientists to get their products to the clinic. I think that's something that we need to put more effort, like globally, each of us. And I know Stem Cell Network is doing a great job at doing this. Kelly, the fact that you're putting this, the NRC is another example of getting it out in the community. So I think this is very important. The researchers need to understand that the goal is not the paper. The goal is to save lives. And we really want to save lives, and we have the ability now and the technology to do that. So we need to encourage our researchers who are making these great discoveries to put them into practice.
Yeah, great. Well said. Min, you have 30 seconds because we're going to have to wrap up.
Kelly, right? So from my perspective, come from an industry, there's two aspects. I think the biomanufacturing I think it's great and it's grown. But one of the things that costs us from a SME is really the patient trials. And here in Canada, it's very complicated. It's very complex. You know, we can knock down the biomanufacturing side, but we still haven't knocked down the clinical aspect of the patients. How do we get the patient? How do we gather the patient? Again, it's a rare disease. Like a standard clinical trial is complex. A rare disease clinical is much more complex. And I see CPI and I think CCRM or anyone that can associate themselves with in a hospital or a clinician, that's great. I think where the United States thrive is really gathering the patient itself and collecting those data, and then the biomanufacturing. I think it's going to follow through.
Okay, thanks very much, Min. I think it's time for us to wrap up. I want to thank all the panelists for session 2. I think we got to most of the questions. There may still be a few in the chat. Yeah, we might be able to answer offline, but I'd like to invite, I think it's Rusini Weratna who's going to come on for final comments and to close out the session today.
Thank you, Kelly. What great discussion, what great insight, and hope for the future. I really hope that we are leaving you today with a lot of new insight, new perspective, fresh ideas, and perhaps some new collaborations as well. As Kelly mentioned, I'm Rusini Weratna. I'm the program director for the Cell and Gene Therapy Challenge program here at NRC. So as we come to the end of today's symposium, I would first of all like to extend a huge thanks to our speakers, moderators, and the panelists for sharing their expertise and insight. And also, I would like to thank our colleagues at Stem Cell Network, especially John Draper and Fiona Cunningham, for their partnership in developing this program. Their leadership has been really essential to making today's symposium possible and a success. And of course, thank you to all the attendees for joining us virtually and contributing with questions and engaging in the dialogue and helping making this symposium a success. So after the close of this meeting, you will receive a survey in your inbox about the event. We would really appreciate if you take a few moments to complete it and provide us with your feedback, which will be really helpful for us to improve our future symposia and ensure that we continue to meet the needs of this community and help the innovation of cell and gene therapies to bring hope to patients and families that desperately need this therapy. So thank you again to everyone.